Dobry wieczór. Nazywam się Krzysztof Szade. Mam przyjemność zaprosić Państwa dzisiaj na kolejny wykład na festiwalu Copernicus. Dzisiaj wykład wygłosi profesor Irving Weissman z Uniwersytetu Stanforda. Na wstępie chciałbym poinformować, że od tego momentu całe wydarzenie, jak i wykład odbędzie się w języku angielskim. Natomiast będą polskie napisy z tłumaczeniem. So, let me welcome you again. My name is Krzysztof Szade, and it is great pleasure to me to invite you for the next lecture during the Copernicus Festival. Today we'll hear Professor Irving Weissman from Stanford University, and the topic of his talk will be stem cells and the future of humanity. Let me shortly introduce you Professor Weissman and his work. So imagine that in our bodies, even 1 million of new blood cells are produced each second. All of these new blood cells are derived from blood-forming stem cells. And while we are talking about time during this festival, these blood-forming stem cells are really long-lived. They produce our blood for whole our lives. Now, Professor Weissman was the first person that prospectively isolated blood-forming stem cells, first in mouse and then in humans. He was the first person that purified blood-forming stem cells and transplanted them in human. Not only he investigated the stem cells, but also during the next years of his research, his group showed how the blood-forming stem cells differentiate and produce mature blood cells. That's why he's often called the father of hematopoiesis, so the process of blood forming. But please don't be misled by this term. Professor Weissman not only made critical contribution to the biology of blood-forming stem cells, but his findings was profound to understand the biology of different kind of stem cells. Now, what is important, he always and constantly pushed to translate his basic findings about the biology of stem cells and to the clinical application, to find the way to translate his knowledge and our knowledge toward the patient bed. And getting back to our main topic of the festival, the time. So there is also a dark side of this longevity of the blood-forming stem cells. If they live so long, they have time to accumulate mutations. And accumulating mutations may lead to blood cancers and blood diseases. So Professor Weissman and his group was one of the first that show how the leukemia, the blood cancers, develop from hematopoietic stem cells. And he also find how to treat this leukemia. He proposed a new method to trigger the macrophages, the kind of blood stem cells we have, to simply eat up the cancer cells. And Professor Weissman is, of course, the world leader in the field of stem cells. But what is equally important, he's well known to be a great mentor. And he trained several scientists that now, with their own groups, make important contribution to the biology of stem cells. And I was privileged to be a postdoctoral fellow of Dr. Weissman. And it was a great time for me. It was a time of great scientific adventure. And I'm sure that today, with this lecture, you will for sure taste at least part of this scientific adventure. So the lecture was previously recorded, but after the lecture, we'll have live call with Professor Weissman. So there will be a great opportunity to discuss with him, to ask questions. So I encourage you to post your question on YouTube channel. So, ladies and gentlemen, let's start the lecture by Irving Weissman.
Well, thank you for inviting me to the Copernicus Symposium. I'm especially proud because part of my family did come from Poland long time ago. Now, the topic I'm going to talk about is stem cells and what the prospective isolation of stem cells can lead to scientifically and clinically. 32 years ago, we published the first prospective isolation of mouse blood-forming stem cells, shown in the picture here. And as we developed the method and followed their work, we found that they were only made one time in development at the embryo to fetal boundary. And therefore, all the blood-forming stem cells you have were born then, and they maintain the system for life. And when things go wrong, like leukemia or blood diseases like myelodysplastic syndrome, it turns out that the diseases start and are important because they begin in the blood-forming stem cells. Years ago, when we used the fluorescence-activated cell sorter to sort cells from the bone marrow of mice to test for which cells could save lethally irradiated mice and self-renew, and then in humans, we found markers that were on stem cells and other markers that were on the other cells, but not stem cells. So you could make antibodies to negatively select those markers and others to positively select those markers. When we were successful at doing that, we went back to some of the practices of bone marrow transplantation, which unfortunately people now call hematopoietic stem cell transplantation. It would be fortunate if they actually transplanted only pure stem cells, but they changed the name. So if this is the bone marrow or the mobilized blood or the cord blood, the source of stem cells, our job is to find the rare blood-forming stem cells and purify them free of any other cells that are there. We found at the company I began, Systemics, that if you have metastatic breast cancer or non-Hodgkin's lymphoma or multiple myeloma, the cancer cells are also contaminating quite often, almost always, the bone marrow or what's called mobilized blood. So you must do sorting, at least positive sorting with more than one antibody for the stem cells and negative sorting for the other, to get pure blood-forming stem cells for transplantation. And we found that those populations, human cells, were 250,000-fold depleted of either cancer cells or T cells. And that meant that we could begin to think of cancer-free transplants. Now, one of the most important clinical trials that was going on in the 1990s and early 2000s was in women with metastatic breast cancer to increase the dose of their myeloablative and anti-cancer drugs to the point that they wipe out the blood-forming system. So people were taking the mobilized blood, freezing it down, treating the patient with what would be a lethal dose of combination chemotherapy and rescuing them. But as we showed, there were cancer cells in the graft. So we knew that that was dangerous and we needed to change the practice. And now, 24 years after the trial began, 8 years since we published this, I can show you the overall survival. So these are women with metastatic breast cancer. They represent all the types of breast cancer, including triple negative. And they got a lethal dose of cisplatin, Cytoxan, and carmoustin. And then they were rescued. First, the standard of care at the time with mobilized blood, which did have cancer cells that could contaminate the stem cells, but another smaller group, but significant group, that had stem cells only. So this is the Kaplan-Meier plot of those rescued with mobilized blood, peripheral blood. Their median survival was about 26 to 28 months. Their overall survival at that time was 7%, now it's 0. The patients who were rescued with cancer-free stem cells, so we didn't put cancer back in their body, dramatically extended the extrapolated median survival to 120 months or 10 years. And 1/3 of them today are alive, at least 4 of the 5 that I'm representing here, without disease. So you would say, okay, that's a therapy. We published that, the beginnings of it in 2000 and this, but the problem was that the company I started was purchased by another company that wished to carry it forward. And then not long after the purchase, in a business decision, decided to spend their money on something else. So that's why this is not a therapy. But if you just look at that and the level of contamination that we'd seen in the original mobilized blood, the amount that could reduce a 10-year survival to 2 years, there were probably no more than 100,000 breast cancer cells in the body just at the end of the chemotherapy. Whereas, as you know, to have clinically significant disease, you probably have between 1 and 10 billion cancer cells before the chemotherapy. So this was an incredibly effective therapy, but the follow-on just with that treatment alone or anything else didn't follow, not because it didn't work, but because of how we do things. So getting back to stem cell biology to see what we can learn about stem cells, Debashi Sahu and Jun Seta, David Dill and I had developed an algorithm that allows you to look at gene expression in any particular set of cells like hematopoietic stem cells or multipotent progenitors or common lymphocyte progenitors, granulocyte, monocyte, megakaryocyte, erythrocyte. What you're looking at is we typed in the names of genes and out came these colors from array analyses. And what you could see is of all the cells in the bone marrow and the bone marrow stroma, HOXB5 was unique and it was expressed at high levels, mainly in hematopoietic stem cells. So we made HOXB5 triple M cherry mice. And when we look at those homozygotes, you could see that of the cells that I had showed you had the phenotype of hematopoietic stem cells, somewhere between 20 and 40% were HOXB5 high or low. The rest were HOXB5 negative. When we did limit transplants of the high or low or negative, the negatives gave multipotent reconstitution at a low level, but on serial transplant, it failed. So they did not self-renew. HOXB5 low gave multipotent, and on serial transplant they worked, but they were not on a per cell basis as effective as the HOXB5 high. And these are just the actual plots of the reconstitution looking at different cell types. So now we know, and this is James Chen, Masanori Miyanishi, Hiro Nakauchi in our lab. So now we know HOXB5 is a candidate for the stem cell niche. Here is the bone marrow niche where we used an antibody to endothelial cells given intravenously, a green labeled antibody, 30 minutes before sacrifice. You can see all the blood vessels. Now, before our study, if you read the textbooks, you would expect hematopoietic stem cells all to be sitting next to the endosteum. But what you can see with these little red dots, I hope, and a high power of it, is that none of them were next to the bone. None were next to the arteries. They were all on the abluminal surface, that's within the bone marrow contact surface, of bone marrow sinusoids and or the blood vessels leading into or out of them. There were only 100 of them per mouse femur, and they represented mainly resting cells as we had seen phenotypically, but about 5% are in cycle. Just observationally, we did not see doublets anywhere. So that informed us that the niche, which is a cartoon here that I drew a long, long time ago, might be restricted to holding only one stem cell at a time, and that we would have to look at other niches for the maturation potential of other cell types. So one of the important points that we wish to know, and which we have tested, do stem cells naturally recirculate from the bone marrow into the blood and then into another bone marrow? We tested this with parabiosis way back in 2001, Doug Wright, Amy Wages, and I, where we sewed together mice. We allowed them to develop a cross circulation. And then after 7 weeks, we separated them and waited for the short-term cells to go away. And to make a long story short, we always found stem cells of one in the bone marrow of the other. And that now is true for the HOXB5 mCherry that Gunsagar Galati and I have looked at. So that means that hematopoietic stem cells at a low rate can leave one bone marrow, go to the blood, and go into another bone marrow. I'm not going to talk about the molecules that do it, but just to say it can happen. So that opens the possibility that stem cells can compete with other stem cells for bone marrow niches. When we developed, all of these people developed in the lab, the hierarchy from blood-forming stem cells to blood, the very first step goes from a cell that self-renews, this is the markers for human, this is for mouse, to a cell that has the same developmental potential, called the short-term stem cell or the multipotent progenitor, but it can't self-renew. So when we transferred these cells, they made all the blood cell types, but it went away in 8 to 12 weeks. But even a single stem cell put in a situation that it's not necessary by itself for the survival of the animal will not only self-renew and give rise to on the order of 30,000 stem cells a few months later, but also all of these blood cells. Because we have each of those cells, as I said, you can do gene expression analyses of them to begin to understand what's going on in hematopoiesis. One of the early things that we noticed, mainly led by Derek Rossi and David Breider, is that the stem cells in the bone marrow of young adults are at lower frequency and have a different potential than the massive stem cells that are in older animals. And so you not only increase 2 to 3 to 4-fold the number of phenotypic stem cells, but you can transplant them, and these will give rise to long-term lymphoid and myeloid, whereas these will give rise to myeloid bias. And when you look at the gene expression at bulk sorting then and now single cell sorting, we see that they have very different gene expression programs, and these are myeloid bias. They have more myeloid transcription factors and fewer than these. And just to make this story complete, the 17 highest myeloid transcription factors or genes upregulated in old over young in mice are known oncogenes in human acute myelogenous leukemia. So you can think about this both as a change of state of the cells, an intrinsic change of fate, and that they do open the possibility for malignant transformation. Krzysztof Szade led a team in my lab that discovered that neogenin-1 subsets the HOXB5 positive young mouse hematopoietic stem cells such that an antibody to neogenin-1 can show that as the mouse gets older, more and more cells are neogenin-1 positive. When you transplant them from young, neogenin-1 negative cells give rise to more lymphoid. I hope you can see that when I'll get rid of this. More lymphoid than myeloid, and the neo-1 positive gave rise to more myeloid than lymphoid. It's not absolute, it's relative. So this is just that myeloid and lymphoid again. And we have some preliminary evidence that it's not such a perfect marker for staining human hematopoietic stem cells, although the drop in the level between lymphoid bias multipotent progenitor and other multipotent progenitors say that we should be able to search. Now, neogenin-1 is a guidance molecule for neuron axon tracking. And it has both receptors that allow the movement toward or the movement away, netrin versus RBD. Just to say that this is reused in the hematopoietic system, and I hope Krzysztof solves the problem of how that actually works. And these are just the ratios. Before we found out about neogenin-1, Wendy Peng, Stan Schreier, and I, the late Stan Schreier and I, found that if you collect bones from old people who have shoulders, hips, knees replaced, and isolate their cells and ask what is their biological potential of the phenotypic stem cells, young stem cells make about equal number of myeloid and lymphoid, but in humans, as in mouse, stem cells from old people are biased toward a myeloid ratio. And this is reflected in the fraction of common lymphocyte progenitors using our own markers, which we'll publish soon, also goes down with time. So this is not related to organs like the thymus shrinking and therefore not allowing you to see lymphocyte development. This is quite clearly intrinsic before the cells would ever get to the thymus. So that led us to propose, and I'm proposing again, that cells that dominate in youth are mainly balanced, but they become less frequent, and cells that dominate in aged are rare clones before. We do not know yet if green ones turn into blue, balanced to myeloid, or if these are cell intrinsic programs. We have evidence, Krzysztof has, that it can turn, but we don't know yet the extent to which a young one could turn into an old one. Now, the important point here, given the COVID pandemic, is that most of us and most animals, when this system was developing, had a certain geography where they would meet microbes in their life. And the immune system that would start to respond to the microbes give rise to long-term memory T and B cells that last as long as stem cells. In fact, a subset of the immune responsive memory cells expressing the homing receptors CD62 alpha lymph node, alpha 4 beta 7 for Peyer's patches, are stem cells which, when they're stimulated, they self-renew for more memory cells and differentiate to give rise to effector T and B cells. All of which is to say that before trains, planes, and cars, we had a system developed that you were living on the memory of the antigens and microbes you had seen before, and you now made cells that were much more efficient at making granulocytes and macrophages, which are first-line defenders against infections like staph, pneumococcus, and so on. So that we have a problem, and you notice that in the age of people who get COVID-19, that old people do much less well, and we propose at least one part of that is that their stem cell system is dominated by the clones that are myeloid biased. Way back in the late 1990s, Toshi Miyamoto, Koichi Kashi, and I were able to get bone marrow from patients that had survived bomb in Hiroshima and had developed leukemia. Here is a paper we published in 2000 on the subset of leukemia patients that had AML1 EDO translocations. So we found that the leukemia stem cell, the cell that can transfer the disease to immune deficient mice, and which in vitro gives rise to leukemia blast colonies that have markers of myeloid differentiation, are not in the stem cell, but they are in the multipotent progenitor or short-term stem cell pool. And in all of the same patients, exactly the same translocation breakpoint from AML1 to ETO was always present in a significant fraction of their bone marrow, wherever you would look at the bone marrow. These did not transfer the disease. They transferred hematopoiesis. It may have been a little biased, but they transfer hematopoiesis, and they don't make blast cell colonies. By the way, the vast majority of leukemia blast cells have markers of myeloid, and they're not representative of the small number, which are about 5%, that are the leukemia stem cells. So these data were so striking that we had to understand what was it that made these and not these leukemia stem cell, and why were these beginning mutations always present in the stem cell pool, but not sufficient to make leukemia. We raised the hypothesis that this is stem cells and this is blood, that events like these are the ones that David Traver and I saw in mouse leukemia, independent events that must occur with a very low frequency, any single cell. If you needed all of those events to make a leukemia, there's not a chance on earth that any single cell would even get 2 of the mutations at once. So we propose that those mutations which could occur in any blood cell with equal frequency as the stem cell will be gone with the lifespan of that cell. But that one that lands for the unlucky person in a hematopoietic stem cell clone, it has the chance to accumulate over many, many years some of the other mutations. If that hypothesis was correct, we ought to be able to get the leukemia stem cells that say have 7 events. We ought to be able to find a cell with 6, another with 5-4, and we ought to be able to phenotype them to see, are they lineage restricted, oligopotent, multipotent, and or stem cells. So that was the hypothesis. So when we look at these, we began by sequencing Ravi Majeti, Steve Quake, Max Jen, especially Ryan Corses, sequencing particular patient leukemias. We did exome sequencing the leukemia and T cells from the same patient because most of them are memory cells that were born before the events leading to leukemia. This is patient number 70 at Stanford. These are all the mutations we saw that were exomic. Some of them in red are mutations that come up over and over in leukemia. So we were able to make DNA primers for each of the mutations from patient 70, isolate single hematopoietic stem cells from the same patient, which, as you remember, can't transfer the leukemia, and then ask whether that clone has this mutation, this one, this one, this one. Now, we could expand those cells a little bit without changing their mutation, but it always changed their epigenetics, so we could only look for mutations. When we did, this patient had, as a first mutation, a loss of function TET2. The second mutation, for sure, was the other allele of TET2, so both alleles are lost. The third was CTCF, the positioning of the transcription complex on the DNA. The mutation changed the DNA binding site for CTCF. And the last mutation, and this and every other FLT3 internal tandem repeat, a receptor tyrosine kinase that does not need a cytokine to trigger through the JAK-STAT pathway, is the last one. Along with the early ones, along with TET2, was DNMT3A, IDH1 and 2, NPM1. So all of those early mutations are changing the epigenetic possibility of the daughter cells of that cell. DNMT3A is needed to close open chromatin. TET2 and oxygen-free radicals generated by the mutant IDH1 and 2 are required to demethylate cytosine and DNA to allow opening of chromatin. Now, this is a general problem, but what over periods of time, it does allow the generation of the leukemia. And just like FLT3 ITD, the activation of the beta-catenin in this transition to myeloid blast crisis, Katrina Jameson and I showed, is the last event. And any RAS mutations, it's the last event. So there is a general hypothesis here of what is happening. As we went through all of these events, and people from the lab began looking at other things, Wendy Pang not only looked at the aging of hematopoietic stem cells, but she showed with chromosome-marked myelodysplastic syndrome that over 98% of the stem cells in every patient that we examined came from a single stem cell that was either monosomy 7 or 5q minus or ribosomal protein mutation. So you can get a clone that takes over all the bone marrow. And Sid Jaiswal, working with Ben Ebert, not with me, after he left the lab, found that all of us have a certain predilection to develop clones of hematopoietic stem cells. And those clones expand, and you don't know what the disease potential is, except if they could go on, if you live forever, likely these are the next wings. But these are thousands of fold more frequent than the actual leukemias or the myelodysplastic syndrome. And Sid has gone on to show, oddly, that these patients with clonal hematopoiesis not only have a high incidence of myeloproliferative and leukemic disorders, but it is the second highest risk factor for getting atherosclerotic diseases. We'll get back to that. So what I've said, I think it's probably the most important thing I've said, is that in AML, the pre-leukemic progression is in a blood stem cell clone. And because these cells can recirculate, they compete with normal stem cells. We have found no leukemias of which are phenotypic stem cells. They're all at the downstream progeny. And since all other tissues that we know that have been examined carefully have stem cells, have daughter cells, and because mutations don't know that they're in a stem cell in the blood or a stem cell in the brain or a stem cell in the bone, we predict, but it's not yet been tested, that this model will be true in all tissue cancers that regenerate from tissue stem cells. When we looked at the gene expression profile of leukemia stem cells, highly purified, and normal human hematopoietic stem cells, we found in mouse and in human that there was an upregulation in the leukemia stem cells of CD47. We found that we could find and then make antibodies against CD47 that are blocking. The team that did this was Mark Chow, a medical student at the time, Sid Jaiswal, a medical student at the time, Ravi Majeti, and then a whole team of us. So we could block CD47, and we found that that was sufficient to block this as a don't eat me signal, and that allowed the cells that have eat me signals to be phagocytosed. This is highly purified human acute myelogenous leukemia stem cells that we stain green with a dye we put together with human macrophages. And we could show that although other antibodies of the same isotype can bind the cells, only anti-CD47 blocking antibodies will lead to the phagocytosis. And that's true of this one, That's true. This blocking antibody, but another antibody that binds CD47 but does not block its interaction with the anti-phagocytic receptor on macrophages called SIRP alpha. If they can't block it, you don't get phagocytosis. And normal hematopoietic stem cells and normal bone marrow are not eaten. And we now know because they lack the eat me signal. So this gave us the idea that we could move to a clinical protocol where we should reduce the risk because you're blocking the don't eat me signal, which can be on both normal and neoplastic cells. But if you don't have the eat me signal, like normal cells, you shouldn't lose it. So here was the first test in leukemia in mice. 3 weeks of treating with an anti-CD47 antibody. 8 out of 9 animals were completely cleared. This is at 2 weeks of the human primary patient leukemia stem cells. And 1 out of 9 took a long time. We couldn't, because the leukemia was more aggressive than the macrophages here eating it, the leukemia one, not the others. So we took this to clinical trials, and it was done with a company I co-founded called 47 Inc. The company is gone now. It's been purchased by a large company. I don't own stock in the large company, so I guess I'm not so much in a conflict of interest. But in the clinical trial done with groups in the US and the UK, we found that anti-CD47 alone in patients with myelodysplastic syndrome with high risk factors, or in elderly patients as a first-line therapy with AML, alone didn't do very much. But if you combine it with azacitidine, which increased the eat me signal that we could understand in the disease cells, virtually all of the patients end up going into remission, many of them so far in very long-term remissions. When we looked at other drugs that can fight cancer, like rituximab, an anti-CD20 antibody, Mark Chow and Asha Lizadeh and Ravi noticed that the rituximab antibody is a humanized IgG1, and that binds to the phagocytic receptor, the high affinity Fc receptor on the immunoglobulin. So we thought, even though the Fc receptor is also on NK cells, that perhaps we could synergize blocking eating and adding much more eating to treat large cell lymphoma. And Ranjana Advani and others carried out that clinical trial at Stanford in relapsed refractory patients with DLBCL follicular lymphoma. About half of the patients go into another remission. And by the swimmer plot, it looks like many of those also are long-lived. So that says already, without a doubt, this is an immune checkpoint, and the immune checkpoint is working through a don't eat me signal. All cancers express CD47 as at least one of their don't eat me signals. They all work through SIRP alpha, which has what's called an ITIM motif that activates SHP1 phosphatase to dephosphorylate the cytoskeleton. So a macrophage, that has signaling from a don't eat me signal on an adjacent cell it's looking at can't rearrange its musculature to engulf and eat and kill it. But we found that the most prominent eat me signal on the cancer cells was calreticulin. Studies by Mark Chow, Sid Jaiswal, Rachel Suganaba Weissman, and I. So now we've got this phenomenon. But calreticulin is not a cell surface protein. It doesn't have a transmembrane sequence. It doesn't have a GPI link sequence. So we needed to understand how could calreticulin be on the surface of the target cell so that it could use LRP1 or CD91 to phagocytose the cells that are decorated with this, and maybe you block the CD47. So that led to experiments mainly carried out in the beginning by Mingye Feng, Jens Volkmer, and I, where we found that macrophages can be the source of calreticulin, and that there is a pathway of TLR receptor agonism stimulation, and we did 3, 4, and 7, where going through an enzyme with the properties of BTK, phosphorylate calreticulin, and as I'll show you, it gets cleaved and released. When we treated the macrophages with poly-IC or LPS or imiquimod, all TLR agonists, it's amazing how high the phagocytic index goes in vitro. Now, Chris Marjan and Mingye and Alison Benuelos have put macrophages in a Boyden chamber across from a millipore filter that prevents cells from going across, but allows molecules to go free. And we had target cells, and we found that the macrophage biosynthesized calreticulin, and when you stimulate it with LPS, secrete it, and it ends up on the surface of the target cells, which correlates with their ability to be eaten. And when with Denon Wang, we screened glycan libraries because calreticulin is a chaperone for growing glycoproteins in the ER and Golgi, it gets released from its KDEL receptor. We found that the asialloglycans bound our soluble calreticulin with very high affinity, and that some tumors, maybe all tumors, elaborate neuraminases that change themselves from sialloglycans on their surface in general to asialloglycans. So the model here is that calreticulin in the macrophage gets activated through a BTK-dependent pathway. It gets secreted and it binds to asialloglycan. With Balanzara, we now know the soluble form and the form that binds to the tumor cells, enabling eating and killing of the tumor cells, lacks the KDEL sequence. So we're getting at the mechanism, how it works. Now I want to take us back to breast cancer, because we found with the help of Mike Clark and friend Sheeran that we could transplant human primary, and this is luminal A breast cancer, into the mammary gland of immune deficient mice, let them grow, and then treat them with blocking or non-blocking anti-47, and blocking can lead to the elimination of the tumor. Paula Bettenker and I looked at a number of these, and we wanted to say, why was CD47 upregulated? And here, Jurkat, a T-cell lymphoma, a particular enhancer, went from being a regular enhancer to a super enhancer, precipitable with antibody to H3K27 acetyl. But in MCF7 and several primary luminal A breast cancers, this one became a super enhancer. And by sequencing, we found this had an NF-kappa B motif in it. And if you eliminated the motif or scrambled it in any way, you didn't have a super enhancer form. Well, if you just step back, this is an epigenetic step inherited by the daughter cells to have expression in the cell it started with and all the daughter cells of high levels of CD47. And it might be different for different tumors. So this we need to look at. But I want you to realize that amongst the most aggressive tumors are inflammatory breast cancers. And what Paula Bettenker and I have shown is that breast cancer cells, like the ones we study, can have TNF-alpha receptors. If you add TNF-alpha to it, you develop not only the super enhancer we're talking, but you also open up the transcription start site as well. So this is an important opening. And I'll just remind you, the late Rich Prain not only was the very first person to show that mice can be immunized against their own cancer and that it's a T cell or cell-mediated immunity that does it, but he also showed that some T cells can help tumors to grow. And this is what I'm most afraid of, is the inflammatory response provides TNF-alpha, the cell upregulates CD47, on it goes. Second point I'll make is that if you combine Herceptin, trastuzumab, also an IgG1 antibody, with the anti-CD47, the anti-CD47 causes stasis of the tumor. You stop it, they grow. Trastuzumab, a little better, you stop it, they grow. But at least in these experiments, they don't. And Razi Upton, Mark Pegram at our group, hopefully we'll get that published soon. Now, it turns out there are other don't eat me signals, and I don't have time to go through them in the detail. I'll just say that we found that PD-L1 turns out to be a don't eat me signal, that a blocking antibody to PD-L1 can lead to the PD-1 positive macrophages in the tumor to have increased eating and in vivo therapy. And that if you add anti-CD47, they seem to be at least additive. That was Sidney Gordner's work. A mirror bar called Roy Moat. And of course, I showed that MHC class 1 also can be, the beta-2 microglobulin can be a don't eat me signal on the tumor for LILRB1 in the as the macrophage receptor. So we now know there are at least 47 PD-L1, beta-2 microglobulin, and CD24 that are candidate receptors, all share the ITIM motifs, and therefore all could be involved in cancer. This just shows that one that we recently discovered, anti-CD24 with Sig-Lectin as the receptor, can be an effective prophagocytic signal, and that too is additive or synergistic with anti-47 on the same tumors. Ovarian cancer, triple-negative breast cancers are most often CD24 positive. And so it says that we must be looking toward precision medicine eventually, where every patient's tumor will be tested for the array of don't eat me signals and the array of available blockers of those don't eat me signals. Now, I know time is getting short, but let me just try to move on some things. As I said, mutations can occur in any cell in the body. And you can have cells that are very dangerous. So years ago, we wanted to look at whether a pathogen infecting a cell could protect the cell from phagocytosis by upregulating CD47. And that was led by Michal Tal, Laughing Bear, Torres, Dolgoff in my lab with others. So that's Michal. And we tested with Kim Hazenkrook's group and others in mice, friend virus infection, LCMV infection, lacrosse virus. And we found it was always upregulated in infected cells, even in malaria-infected cells. And that antibodies that block the CD47 have some therapeutic effect, and that this does lead to cross-presentation in friend and LCMV to stimulate T cells to keep acting against the persistently infected cells. We found that was true in samples from the lungs of patient with ARDS, this pandemic. So it's part of the problem. It's true in Lyme disease and in Salmonella. With Carolyn Stoddart, Kim Hasenkrug, Laughing Bear, Michael, Laura Myers, and Mike McCune, his name got dropped somewhere. We found that SCID mice having human fetal tissue, immune deficient mice, can be infected with HIV. We showed that in 1988. It has been a testing ground for drug ever since. And that when we treated those animals with pathogenic HIV, we could dramatically reduce the viral load. Now, we did increase the CD4 to CD8 ratio in them. When we decided to repeat and expand this study, we were banned from doing that by an executive order from the US White House. So we can go to jail if we try to repeat this study. So quite clearly, the NIH is not just been ordered to advance medicine for the health of people, but it also now takes on political and religious overtones, which I've written about elsewhere. So what we said now is all cancers, all infectious diseases can use the CD47 to protect the pathogenic cells from phagocytosis. Gerlinde Wernig and I developed a model of fibrosis, idiopathic pulmonary fibrosis in this, but non-alcoholic steatosis, that is fatty liver fibrosis, renal fibrosis, scleroderma, so on. I won't go into the model except to say here's how the lung looks normally. When you turn on this disease, the fibroblast precursors, which Yuval Rinkovich and I showed, come from the mesothelium of the pleura covering the lung, grow inward as fibroblasts. They set up a huge inflammatory reaction, including IL-6, IL-1, and so on, and you have a lung that looks like that. The current drugs barely treat it. The current drugs plus anti-CD47 treat that mouse model a little bit, but if we add anti-inflammatory and that, we can largely restore that development. That's adhesion. That's a fibrosis. Here's another fibrotic condition, surgical adhesions. Everybody knows about this who is a surgeon. And I'll just make a long story short. Jonathan Yuval Rinkovich and I have shown that it's the same mesothelial surface that, if it becomes ischemic, can send the fibroblasts out into the peritoneal cavity where they induced a neutrophil inflammatory infiltrate, and the mesothelial cells re-express fetal mesothelin. So now these are the complete engulfment in an animal of an induced adhesion by ischemia. After it's gotten to that state, you can treat it with anti-mesothelin or anti-mesothelin and anti-mouse CD47, and you can completely clear or nearly completely clear that adhesion. So we looked at other diseases. Nick Leeper began showing that human atherosclerotic foci overexpressed CD47 and calreticulin in the atherosclerotic focus, and that in an animal model of atherosclerosis, ApoE, knockout, high-fat diet, that you can prevent the atherosclerosis with that. When you use the rainbow mouse to ask what's the clonal representation of the smooth muscle cells that are blocking the arteries, you can see large blue or large orange or large green, single cell clones that form the atherosclerotic plaques. and not yet published in patients receiving anti-CD47. Nick and the team have shown that patients do get a reduction of plaques in the carotid that have gone through the therapy. Now, if I have 5 more minutes, I'd like to finish this because I don't want to leave you with the idea that we're ignoring why I got into this. Being able to do hematopoietic stem cell transplants safely between donors and hosts, or autologous cancer-free stem cell transplants. So you all know that an allogeneic transplant, the bone marrow in mobilized blood or mobilized blood contain T cells, which are entirely or almost entirely responsible for the pathological graft versus host disease, and that it's a miracle of the art of bone marrow transplant medicine that you can give enough immunosuppression so that the patient's neoplasm might be destroyed. But if you want to use stem cell transplantation to regenerate genetically defective or iatrogenically depleted stem cells, so you got to go from a donor to a host. The T cells are not doing anything to help you at all. So we demonstrated about 40 years ago almost, that you can induce tolerance to donor antigens by making radiation chimeras. In those animals, for example, if they're diabetic nods, you can go from getting spontaneous type 1 autoimmune diabetes. If you now irradiate them and give them antibodies to some T cells, but give back their own bone marrow, you buy a month. That's as far as the bone marrow transplant community has gone, even though we published what I'm going to show you next in 2001. If you purify the stem cells so you're not giving back autoimmune T cells, you buy another month. But if you take stem cells from a donor that is not genetically prone to type 1 diabetes by allowing clones of T cells to escape thymic and post-thymic selection, like in BLG7, so that would be fully matched but otherwise different mice, you prevent the disease, and if you co-transplant stem cells and islets, long after the disease, you cure the disease. 2001, not a single patient has been treated. Same works in lupus, although we don't need a graft, we just need to stop the autoimmune attack. Work done by Julie Christensen, Stephanie Smith-Burden, and I at a company called Celera. You can block it. But irradiation and chemotherapy are too toxic for kids who come in drinking and being too much, too toxic for old people with autoimmune diseases. So we decided we would try to substitute for radiation or chemo antibodies that will block stem cell functions. So we got a function-blocking anti-C kit from Shinichi Nishikawa. Koichi Ikuda and I showed years before that all hematopoietic stem cells express and need to express C-kit. And one dose that Anishka Chekovitz, Deepta Bhattacharya, and Dan Kraft did. Anishka was a student at the time. It was her idea to go forward with this. You increase a single transplant's donor chimerism with just anti-C-kit in a skid mouse and 5,000 stem cells added. But when we tried this in immune-competent animals, instead of depleting many logs of stem cells, you got a mild depletion. So we needed to go on and do something else. Well, we knew that we'd already proven the principle that anti-CD47 could synergize with another antibody so long as it was IgG1 in human or IgG2A or 2B in mouse, as this anti-C kit was. So we said, maybe we can synergistically and selectively remove hematopoietic stem cells with no radiation. And this is a syngeneic transplant. We gave anti-C kit, anti-CD47, transplanted syngeneic hematopoietic stem cells that we had marked. And that, and only that, leads to engraftment. if we now move from that to HLA match, but in the mouse model of it, now we have to get rid of T cells as well as stem cells, because T cells in a match graft are the main resistance. So you add those antibodies together with anti-47, and if you don't add anti-47, you don't get donor chimerism, but if you do, you get multi-lineage chimerism. And finally, Benson George and I went to a much more frequent. And the reason we had to is we realized, as the transplanters had realized, nowadays 2 children is about the max you have in a family. So you don't have that 25% chance anymore of a full HLA match, except rarely. That's just modern times. So we wanted to go to haploid transplants, AKR by black into BAL by black, a shared black allele. And we did the anti-C kit, anti-T cell. We had to add anti-NK cell, anti-CD47, and blocking antibodies to CD40 ligand, but then we could get haploid transplants to succeed. And we proved they were immunologically tolerant now for 2 years. Now we have to sacrifice the animal. Not only are they chimeric, but a transplant from the stem cell donor is accepted, a heart transplant in the ear, whereas the third party is rejected by 13 days. So this is my final slide. We think that by this aspect of studying stem cell biology, we can get rid of radiation and cytotoxic drugs. We can remove the immune response elements with antibodies, all of which we now are taking into the clinic. The anti-C kit has been done in SCID children with a study led by Judy Shizuru, Rajni and Maria Grazia Roncarello protocol that we helped write. And they're engrafting with anti-C kit alone in a SCID. But eventually, we'll want to be able to put organs or organ stem cells along with hematopoietic stem cells from the same donor. Now, if you have a donor that both are available, you could do it. But that made us think, and that's why we have a stem cell institute, that if we could embryonic stem cells or induce pluripotent stem cells to give rise to blood-forming stem cells, and for example, like we've studied, brain stem cells for neurodegenerative diseases, spinal cord injury, macular degeneration, that this could be off-the-shelf antibody conditioning and stem cells from at least partially matched donors. So I know I've taken probably over my time, but I want to thank you for your patience. I want to thank the inviters of this to my meeting, especially Krzysztof Szade, who helped revolutionize my lab by thinking out of the box of where we could go to look for molecules that we could now use to go forward. And thank my support, which is the California Institute of Regenerative Medicine, the Ludwig Cancer, Siebel Stem Cell Institute, a couple of anonymous donors, and for many, but not all studies, the National Institutes of Health. Thank you.
Welcome back again. We have a live connection from California with Professor Weissman, and we will have a chance to ask questions and discuss with Professor Weissman. So I already saw there are some questions on our chat here. There is still time to ask more questions. Dear Irv, good afternoon, or rather good morning, because it's morning time in California, I guess, right now.
Good morning.
Yes, how are you? We have a big online audience here, and we just again would like to thank you for the lecture, and it's great to having you here live with us. So I will start with some questions here that you already asked. So the first question was about the mutations in leukemia from Albert Sosnowski. Since we know what mutation in stem cells are causing leukemia? Can you use CRISPR and Cas9 method, possibly used in the leukemia treatment and the fetal stage?
So you could use CRISPR to fix the mutations, but you would have to do it for every cell in the body to get rid of the leukemia. And not even CRISPR is that good. And certainly the vectors that one uses to bring the CRISPR constructs in don't know how to go to every cell in the body. If you put a lymphocyte in the body, it needs many homing receptors to go from the blood to a different place. Imagine now how you could get a virus to do what a lymphocyte does. So I think it's important diagnostically, it's important to figure out what therapeutic targets you could do something with. But as we said, at least for leukemia and perhaps pre-leukemic changes, the anti-CD47 plus azacitidine seems to clear up both the leukemia cells in elderly people with first onset leukemia, but also myelodysplastic syndrome, which has got the pre-leukemic phases, but the patients won't necessarily go to leukemia. So I think it's very promising that anti-47 plus azacitidine works. And now we have to see, does it work in other myeloproliferative neoplasms? And maybe does it work in clonal hematopoiesis? So even though you're healthy with clonal hematopoiesis, I think there's something you would like to do to get rid of those cells.
Of course. Yes, thank you. So we have the next question from Monika Pasternak. What is the perspective in treating patients after stroke with Ips cells? I was reading that this is effective in rats. What is your view on this topic?
Well, there are people who have taken ES or Ips cells and generated not pure, but hematopoietic progenitors. But we've found, Kyle Lowe and I and others, that the hematopoietic stem or progenitor cells you have are at the stage of the yolk sac blood island. And in at least the mouse and probably in humans, you have to go from the blood island to the fetal liver to become transplantable. So I think all of us working on the field of IPS to make blood-forming stem cells know that there are barriers that we have to overcome. But because we have the precise definition of the fetal liver, the yolk sac stage and the fetal liver stage, I think it's possible we'll learn how to do it. And that will change all of bone marrow transplantation, because you won't lack for donors. You could make many, many different donors. And you might also be able to make, as we're testing now, that you could make brain stem cells. So if somebody had a spinal cord injury and you needed to have brain stem cells, but you did not think they could survive the immunosuppression, like an organ graft, you would want to co-transplant blood-forming stem cells and brain-forming stem cells from the same embryonic stem cell or IPS line. So we see great potential, and I hope it will move faster than it has. But at least I am pretty confident it's not an insurmountable problem.
Yes, let's hope it will get finally to the patient bed. So let me ask the next question by Katarzyna Mleczko-Sanecka. Is there any possibility that environmental factors like caloric restriction or dietary manipulation or exercise may influence a ratio between lymphoid and myeloid-skewed HSCs during aging?
Well, it's always possible that environmental factors will do that, but I don't know of any direct connection of a particular lifestyle or diet or exercise or fibers or anything that have changed this. I do want to say that, as you heard in the latter part of my talk, most people think that if you took care of diet, exercise, mood, fiber, you could prevent atherosclerosis. But now we know for sure, atherosclerosis is like a cancer. It is an overproliferation of the precursors of smooth muscle cells. And as I showed one slide going by, it's a clonal amplification, like clonal hematopoiesis, but clonal smooth muscle cell generation. And the defect in that clone causes inflammation, and that upregulates CD47 on that clone. So all of this now brings us down a scientific line to deal with atherosclerosis that no diet, no exercise, no change of your mood, no belief in this or that will change. So I prefer myself to follow the scientific line step by step, then hope that there is such a thing as a healthy lifestyle that would prevent the disease.
Yes, thank you. And let's move to the next question by Justyna Sośniak. Is there any currently available drug on therapy, at least at test, for patients suffering from pulmonary fibrosis using CD47 antibody?
It's a great question. We have not developed a test. I'll just say Gerlinda Vernig has looked at many biopsies or autopsies of lungs from people with fatal pulmonary fibrosis, and all of them stain the fibroblasts and other cells in the lung with anti-47. But knowing that you can stain a tissue biopsy, which is dangerous by itself, and inventing an image test that wouldn't be dangerous, there's something in between. So I think that one could use CD47 related reagents that penetrate and that might have MRI or PET scan probes attached to them. So I think it's possible, and it would be valuable, especially in the early phases of pulmonary fibrosis, when you might be able to reverse the process or the inflammatory process and help save the patients.
Yes, let's hope it will be possible. Thank you for your answer. Now we have a discussion and a comment from Joseph Dulac, and I will just read it. Dr. Weissman, thank you very much for a very interesting lecture. It's great to know how the knowledge on HSC enlarged since your visit to your Krakow and our university in January 2015. It's a pity that we are not able to welcome you again this year in May, as initially planned, but I believe strongly it will be possible in the future. The bone marrow transplant saved life in many blood diseases, and pure HSC transplants may be even more effective, as you have shown. However, I want to ask you to comment on the situation we are facing now all over the world, in USA, but also in Poland. The claims that the cells isolated from different tissues and incorrectly named mesenchymal stem cells are the miracle treatment for almost any disease and conditions one can imagine. Starting from autism, children paralysis, amyotrophic lateral sclerosis, blindness, and recently even COVID-19. What would be your message to the desperate patient or parents facing such often very expensive offers, but also to the physician and scientists offering them?
In any proposed therapy, and especially stem cell therapy, there are two questions you should ask before you seek treatment or, as a doctor, prescribe treatment. First, in the early phases of the clinical trial with, say, mesenchymal stromal cells, because stem cells, you need to ask the person who oversaw the clinical trial from what we would call an institutional review board, show me the evidence that this is safe. So that's first question. But an institutional review board doesn't ask if it's effective. And that's the most important question if you're going to risk a therapy like that. So then you ask the question, did your government equivalent of the Food and Drug Agency, FDA, in the US, test it objectively, effective and safe? Those are legitimate safety requirements that we know have to be in place. If you think back to the Nuremberg trials of the German Nazi people who did experiments on people without regard to their safety or their lives, it was in response to that we needed to have a way for people not involved in the therapy, not making money from the therapy or having political power because of the therapy. We need to have a way people independent of that to follow the safety of the recipients. If somebody says to you, well, I have mesenchymal stem cells and they work in this or that, and I'll just inject them into you, trust me, the FDA is getting in the way, the government is getting in the way, the hospitals are getting in the way. Don't trust them.
Yes, so this is very important, I guess, just to save the hopes that a lot of people connect with stem cells, and these unproven therapies can just burn out the promise that stem cells can bring to us. Yes, we have the next question from Karolina Bukowska z Krakowa. Is there a possibility that this is a bone marrow niche that changes the bias of HSCs during aging from lymphoid to myeloid bias?
That is a terrific question. And I would say both and I probably say that is open in order for us to understand that, so that one could look at the cells in the niche from young and old and say, do they change with age? Do their signals to stem cells change with age? Because we know you could have inflammation that changes gene expression. TNF alpha could cause NF-kappa B to open up or close certain genes, and now you have an inflamed cell. So I think it's a terrific question. Charles Chan is trying to study it at our place, and we'll wait and see.
So we have a follow-up for this question. Or is it possible that the bias results from the exhaustion of the lymphoid clones with age?
That's another good question. The way one will be able to study that, I think, is to follow the generation of the lymphoid cells. Now, when we first did that, and we did it with humans, we noticed that the common lymphoid progenitors in old people's bone marrow was greatly reduced. So that's before you ever make the mature lymphocytes that could become exhausted. So we know for sure that the myeloid biased hematopoietic stem cells in old people, as in old mice, can't make even the precursors of lymphocytes at the rate that you need. So it is possible, but it would be acting at an earlier stage of development than how we understand exhaustion of mature T and B cells.
Yes, that's a crucial question we still have to answer. Yes, so we have next question by Anton Slivka. To which extent it is possible to block CD47 or in general increase ITMI signal before it becomes destructive for normal cells?
Well, you're asking a very important question. We don't get many normal cells that are lost. As you know from the trials, in fact, the only event we saw when we gave the first mouse, the first non-human primate, and the first human anti-CD47 was an anemia that was dose dependent. The red cells went down. Now, we knew in our studies that if you give a low dose of anti-47 so that your anemia is no more than 5 to 10% drop in hemoglobin or red cell count, that a week later, you can give 90 times that dose and not have any more anemia. It is complicated how that works. One aspect is with the little bit of anemia you would get with the low dose, it turns on erythropoietin in response, and now you have very young red cells that have a lot of CD47 and no eat me signal. So that's part of it. But when we look at the timing and the percent and the events that go on, we're pretty sure that the red cell becomes changed with the first dose, so it's no longer susceptible. If that becomes even safer than it is now, then one could say, is it going to be important to use anti-CD47 early as a first-line therapy? Or even if you had the genetic predilection for a blood-forming disease or myelodysplastic syndrome or clonal hematopoiesis, will that rejuvenate the system by getting rid of the biased cells, leaving the younger, healthier stem cells? We don't know, we're testing it. And luckily in a laboratory with mice, we can do those kinds of tests. I think that people will have to look very much deeper into those cancer patients who are getting anti-CD47 therapies and test as many systems in the body as you can so that you could see if you're improving things that you're not looking at. Do you improve the little fibrotic patches they might have as they age? Do you improve the atherosclerotic? Do you improve the response to vaccines? So will you make a person my age, I'm 80, able to respond to the usual dose of influenza? Because I probably don't respond now. So those are the kinds of parameters that if we can convince the pharmaceutical companies that are doing the clinical trials to do allow much wider testing, so long as it doesn't endanger the clinical trial.
Yes, thank you. For sure, 47 has a huge potential, but of course there are still much more detailed studies that need to be answered to use its full potential in a possibly broad group of patients. So let me finish with my question, kind of a general question, but I think it is important. At least I would like to know your view. You convinced us, at least me, that the stem cell works and the huge potential of regenerative medicine. But do you think it will be ever possible that these stem cell therapies will be done in large clinical scale? That they would not be restricted to selected group of likely rich people, but they will be broadly accessible to the people that can really benefit from it. How is your view? How it will develop?
So it's a very good, complicated question. In the United States, with a highly capitalist system and private insurance, the ability to be treated does depend on how much money you pay for your insurance. So that means you have to change the insurance system to make things broadly available. The second part is to have a realistic look of the cost of developing a therapy so that one could find ways for economies, but also to say to the pharmaceutical or biotech companies, you know, in the first part, you actually made a discovery that was discovered using government research funds to make a discovery. So you're not fair to charge for a discovery you didn't pay for. But now if you look at the realistic cost for you to take it from the discovery to the therapy, then that should help determine the charge. We have an experiment in California called Proposition 71. I helped write it, where the state government could fund a discovery at academic institutions, not only the discovery, but the preclinical proof of principle, the development of a team to develop a clinical trial. We did that with anti-47. We got grants of 20 million and 5 million. And with that money, we filed an IND, Initial New Drug. We did it from academia. No drug company paid for it. No drug company should charge for it. The state has done that to make the transition from discovery to clinical trial and getting rid of the risk that it might be dangerous as its prime responsibility. If we had that as a general means of your state and our state and every state doing it, they can legitimately either share in the returns from the sold product to help fund the research and help fund care for people who can't afford it. We do that with Prop 71. So it means, once again, that you are looking at ways to modify or restructure the existing highly capitalist system in order to bring therapies to people.
Thank you. Yes, hopefully we will be able to, as you said, to work on this transition from the bench to the patient bed without or being as independent as possible from the pharma companies and all the restrictions connected with the business decision. Yes, so if there are no more questions, dear Irv, I would like to thank you again for your great lecture, for connecting with us with this live discussion, for discussing us, for answering the questions. And of course, I would like to thank all our audience. And we hope that your work and the work of other biologists all over the world will make the stem cell therapies possible, and all of us will benefit from it. Once again, thank you all for this lecture.
And thank you for honoring me to allow me to have the lecture. And next time we do it, let's do it in Poland.
It was a great pleasure, and it will be honored to host you in Poland. And as quickly as the pandemic is over, we invite you here.
Good.
Thank you very much. Goodbye.
Bye-bye.
Dobranoc.